but between 12:30 to 1:30. Then after 1:30 we are moving to the campus, TNAU campus. So where you are going to visit the insect museum and organic agriculture, a department which was insisted by our director Sefipra. So we want to expose the people to the organic agriculture department. They are having a trials on natural farming where you can see. Then we can move to our pathology department where you can visit the different laboratories, and we will afterwards we will break. We will come back to the hotel for the dinner. So this is our today's program. So I am reading the names of the persons. If anybody want to advance your presentation, please kindly tell. Dr. Malati, Dr. Sudhini, Dr. Nagendran, Sumia, Dr. Sumia, Dr. Thirumalai Swami, Dr. Harish, Dr. Bharanidharan. Anybody want to present today forenoon itself? Please, you can. Any other person who want to advance your presentation to today's forenoon is welcome. Okay, hopefully we will complete all these 7 presentations by 12:30 or 12:45. So today's tea break will be squeezed to 10 to 15 minutes. Okay. Okay.
Now I request the chairpersons, Dr. Andy Kumar, IIRI New Delhi, and Dr. John Knoll from the France to proceed the seminar.
Somebody should run. Shall we start? Yeah. Sure. Good morning to all of you. At the outset, on behalf of the organizers, I welcome you all. The second day of this session, this program, very impressive program we had yesterday. The whole day, we witnessed very interesting presentations, discussions, and of course the food also. Okay. So we'll continue from what we left yesterday. Already it is communicated to you all. There are 7 presentations already lined up. It's our request, the request of each one of us here. Try to restrict your presentation in 20 minutes so that we can have some discussion. So I believe the monologue from the dais doesn't convey anything unless and until we get feedback from from the audience, and the discussion emerging out of it is very, very important. So it's also my request, like, try to wind up by 15 to 18 minutes, then 2 minutes of discussion is needed. Let's try to figure out. And also equally respecting the speakers who made their slides, spent maybe hours to convey what they have done, what they have been doing. Let's listen from them. So over to my co-chair for his observations and any comments.
Okay. Thank you very much. So we have a very diverse program. However, it will be focusing on diseases. And what is nice is that we will have talks that are dealing with quantitative problems related to yield losses caused by a various range of pathogens, but also we will deal with qualitative losses, which is an important topic too. So maybe in order to be on a good track with the timing, I will set a timer for 18 minutes, if it's okay with you. Okay, so thank you very much. I think we can start from now on with the first. Yeah.
With the permission of my co-chair and all audience, I call upon Dr. P. Malati. Dr. P. Malati is a principal scientist in one of the very old institution in the country, Sugarcane Breeding Institute. It's one of the hundreds of institutes under ICAR, or Indian Council of Agriculture Research. Dr. Malati has done a wonderful work in sugarcane. In particular, a patented technology came from her research program. That is what is called as set treatment, and those technology has been transferred to many companies, industries, and she takes credit for this particular innovation. And she's a pathologist. Let's listen from her what she has been doing now. Yeah, over to you, Dr. Malati.
Dear Chairman, Co-Chairman, Delegates, good morning everyone. First of all, I thank the organizers for having given me this opportunity to share our experience and also the recent trends on the management of sugarcane diseases. I feel proud to say that, it's okay now? Projections? Okay. Okay. I feel proud to say that I am from the Pioneer Institute of Sugarcane Breeding, that is Sugarcane Breeding Institute, which is one of the oldest institute, more than 120 years old, that is which has been established in 1912 under Indian Council of Agricultural Research, which is located just 2 kilometers away from Tamil Nadu Agricultural University. And I think I need not emphasis on the importance of sugarcane, because everybody knows it is a multi-purpose commercial crop, and India stands next to Brazil in its production. And in worldwide, our institute has the highest germplasm collection, and we feel proud of that for developing the varieties for crop improvement. And at the national level, we supporting the breeding program for the whole nation by having the National Hybridization Garden in our institute. So this is the season for hybridization. So because of that, about more than 85% of area under sugarcane cultivation in the nation is occupied by our varieties. To such an important institute, as a plant pathologist, we are supporting the breeders in way of identifying the resistant sources, starting from the breeding program till the release of varieties resistant to major diseases. Next one. Okay, sorry. Here. Coming to sugarcane pathology, actually, so, as far as sugarcane is concerned, more than 250 diseases have been reported worldwide. But as far as India is concerned, about 10 to 15 diseases are important, which are caused by various kinds of organisms, that is fungal, bacterial, viral, and phytoplasm. And however, invariably, all kinds of diseases, that primary source of, actually the planting material serves as the primary source for the spread of all kinds of diseases. Coming to the disease management strategy, we are employing all the principles of disease management, starting from exclusion. That is, under exclusion, we are recommending the tissue culture, that is medicine-derived tissue culture technique for the management of viral diseases. Under eradication, we are recommending the thermotherapy as well as chemotherapy for the management of phytoplasma, bacteria, as well as fungal diseases. Under the protection principle, we are recommending, we have identified effective molecules of chemicals as well as bioagents for the management of all kinds of diseases, including mainly on fungal diseases. This slide shows the important fungal diseases, which include the red rot, wilt, smut, and pocobine, and the top rot. These 4 diseases are called as stalk diseases, while the rust and other leaf spots are foliar diseases. Coming to the bacteria, among the bacterial diseases, the retum stunting is the most important one in India. Then followed by this mycoplasma disease, GST, that's a grassy shoot disease. Then among the viral disease, this mosaic, yellow leaf disease, and recently the leaf wrinkle are the important diseases. Coming to the approaches for disease management, there are 2 main aspects. The first one is identification of resistant sources. Actually, it is mainly covered under the program of All India Coordinated Research Program throughout India, for which we have two ways of identification, that is artificial, that is under controlled condition, as well as in natural conditions. For diseases like red rot, smut, wilt, we have both artificial as well as natural condition for screening program. For other kinds of disease like pocobine, rust, and non-fungal diseases, we have only screening under natural conditions. Then coming to the management approaches, we are approaching under in 2 ways, that is both molecular as well as conventional approach. Under molecular level, we are mainly concentrating on the characterization of potential target genes for the management. And then for regarding in viral pathogens, we are having this RNA silencing as well as genome editing program. In our we are continuing that one. And then for fungal pathogens, we are mainly concentrating on the identification of genes, that is potential target sites by genomic and proteomic approaches, and for pathogens related genes for antifungal, that is for fungicidal management, and the antifungal genes and the defense related genes for the genetic engineering or genome editing approach. Under this molecular approach, I want to just brief about in 2 slides about my experience in the this molecular approach in correlation with the conventional approach. Actually, during this past 20 years, we have actually phylogenetically we differentiated the least virulent and virulent isolates. Then we employed the proteomic and genomic approach, and we identified the potential candidate genes by proteomic approach. And then this is by ditrophic interaction, that is during host pathogen interaction. Then further we also involved tritrophic interaction. Here we involved the antifungal microbes also, and then we identified the potential candidate genes for related to pathogenicity, antifungal nature, everything. Then, but here we found that it was interesting to note that the cytochrome P450 was found to be commonly expressed in the proteomic approach. Then in genomic approach we confirmed under SSH. Then finally, then again under transcriptome approach also we have confirmed, and finally we found that it is found to be the potential target site for azole fungicides. Simultaneously, we were validating these fungicides for conventional management, and we found that it was highly effective for the red dot management. Nowadays, we are recommending the combination of azoles and strobilurins for the management of all fungal diseases. Coming to the technologies, we have 3 important technologies. In the 1970s, we introduced the thermotherapy with the aerated stream therapy or hot air therapy for the management of phytoplasma as well as bacterial diseases. Then in 90s, we have introduced this meristem culture combined with the virus indexing program for the management of viral diseases. Then third, during last decade, we introduced the mechanized priming technology for the management of fungal diseases, involving developing the sugarcane set treatment device. Now I am going to speak much about this technology, this mechanized programming technology, how we developed and how it is useful for, and how we are applying for the management of diseases in sugarcane. Actually, we identified in 90s, we have identified effective molecular fungicides and bioagent and took it for the farmers for the management in the under field condition. But under field conditions, if you see in the farmers condition, they may not go for set treatment, or even if they go for just dipping 15 to 20 minutes or something, they will go for. But our research findings strongly recommend that at least overnight soaking is required for effective infiltration of the chemicals into the planting material. Then we tried for under field condition, but we could not do, because due to considering the voluminous planting material as well as the requirement of more quantity of chemicals, we could not recommend to the farmers. Then we conceptualized an idea of having some mechanized system which is able to deliver rapidly as well as effectively into the planting material, the plant protection chemicals. Then based on that, we have developed a prototype. We fabricated a prototype which works under the principle of vacuum infiltration, and we found that this system, this mechanized system, compared with the overnight soaking, this uptake as well as effectiveness was found to be equal. And then we standardized the vacuum level as well as duration of treatment during under lab conditions. Then we validated this prototype under pot culture conditions for the delivery of fungicides, biofertilizers, inducers, and as well as fungicides, and along with the bacterial biocontrol agents, everything we have validated and found to be highly effective. Then we validated under field condition for the management of red rot in comparison with overnight soaking as well as this mechanism, and it was found to be on par in the effectiveness. And apart from that, there was huge saving of chemical. Then we found its advantage, and we filed a patent in 2011 itself. Then we joined with the nearby engineering institute, ICR Institute, and we fabricated different kinds of devices for the validation, and we validated in our field condition for the management of different kinds of fungal diseases. And we found this system was, and we found that it was on par with the overnight soaking and was highly effective. And we found that this system was found to be simple, and because it is easy to operate, then rapid, it is within 15 to 30 minutes of then it is effective with the uniformity. That's compared to overnight soaking, it has uniform treatment and economical. That is, it requires less quantity, only 1/4, and that also it is eco-friendly. That is, we can have repeated use of the same chemical throughout the day. Then practically feasible and amenable for large scale application. Then able to deliver. Apart from that, it is able to deliver many inputs at a time for the management. Then it is able to protect sugarcane system from both biotic as well as both biotic and abiotic stress. Considering the importance of this system, that sugar mills as well as entrepreneurs come forward and starting adopted this technology. This is the validation under field conditions in sugar factory areas, in different sugar factory for the management of red rot and smut. Here we used 100% infected canes for the trial. And we found that there was a significant reduction in the disease incidence, as well as along with the disease incidence, there was improvement in the yield. We can see this gappiness in the smut infected as compared to single application. Here in the endemic areas, unlike a normal situation, we recommend the secondary delivery of chemicals through drip delivery or spray for the management. Because in normal situation, set treatment alone is enough, but in an endemic situation, we require secondary application, because for the annual crop like sugarcane, it is required. Then apart from chemicals, we validated for the delivery of bioagents. That is, here we validated trichoderma as well as Penibacillus alvi, and the combination of bacterial antagonist with the fungicide, and we found that the combination was found to be highly effective as compared to individual application through this mechanized delivery. So we can see the difference between the inoculated and this one till harvest. Then we demonstrate this technology in both tropical as well as subtropical India. Here you can see this is the demonstration in subtropical India, and this is our wonder variety. One of the variety was broken recently. We got breakdown of resistance for red rot, and we proved this technology for that, and reduction in disease incidence, and also along with improvement in yield. Then this technology has been accepted by ICR, and it was released as one of the technology for integrated management of red rot for the subtropical conditions. Then Apart from disease management, then we thought of having some nursery program, because in the main field condition we are using the bigger size planting material. So farmers felt it difficult to implement this technology for direct planting. So then we thought of having the nursery program. Here we have standardized the combination of different inputs: fungicide, one insecticide, then nutrients. So macro and micronutrients we combined. Here we used the small size sets, that is single bud sets, compared to 2-budded or 3-budded sets. It is so easy to operate. So within one treatment we can cover 1 acre planting. So for direct planting as well as for settling production, we found there was enhanced improvement of germination as well as vigor of the plants. So we is able to select primary settling, that is first quality settlings for the planting. So it is not only improve the vigor here only, in the planting, in the main field, it is able to protect the plants from the infection from for up to 5 months. Then now we are from our institute, now what we are recommending means, first initially we will go for tissue culture raised seedlings from any variety, recommended variety. From that we will raise the seed crop. This seed crop, from this seed crop we will make the settlings and we distribute to the farmers. This is the normal thing in the, from factory, from mills, from our institute to mills and mills to the farmers. This is our normal, this is the settlement practice we are undertaking in our institute for all the experiments. So now this technology also has been recognized at ICAR level. It is being recommended throughout India. Then regarding the dissemination of the technology, we are disseminating the, okay, 5 minutes. We are disseminating the technology by state level program as well as national level program. Under state level, we have one of the program as NADP RKV, and the national level we have ICRIP as well as NFSM program. And at a national level, then under ICRIP, about 13 ICRIP centers in 4 zones, they have confirmed for the past 4 years, they have confirmed, validated and confirmed its efficacy for nursery program as well as for disease management. And under NFSM, we are disseminating the technology. Actually, this technology has been commercialized in 2016 itself. So all the sugar mills have started implementing this technology. So now what we are doing, we are just disseminating the technical inputs for all the sugar mills for follow-up. So at present, more than 500 units have been installed throughout India, among which in subtropical India, like 2 states, that is UP and Bihar, are more important. And here in tropical India, Tamil Nadu is the major one. And now this technology has been, the patent has been awarded in 2024. And here we can see the adoption of this technology by the sugar mills and the entrepreneurs. Then this is about the dissemination of technology in state level. Actually in 2000, when we commercialized the technology, all the private mills started the application, implementing the technology, but in cooperative mills they could not implement. So we got, through support of government of Tamil Nadu, we have supplied the devices to the, to the cooperative mills, and we have disseminated the technology. Based on our inputs, they have recognized the technology. Within 1 year, you won't believe this, 100 entrepreneurs have been developed in 14 mills. So this is the, then also this technology has entered into subsidy. So now the devices are being supplied throughout India under a subsidy. This is with respect to Tamil Nadu. This is, then it is one of the another major sugar bowl is UP state. Here also in all the different factories they have this effectively implementing this technology for seed program. Then this is under another state. Here entrepreneurs have been developed in from 2016 itself in Bihar. Then apart from this, simultaneously we have been working on integrated, we want to develop integrated disease management strategy by utilizing this mechanized priming technology. That is smart delivery of agro-inputs including physical, chemical and biological agents for the management of all kinds of diseases. Sorry, 1 minute. Here we can see this is the normal set treatment device for delivering the bioagents or the chemicals, and this is the upgraded unit for the delivery of hot water. Actually it works only under the vacuum. Here we have combined 3 major things, that is vacuum level, temperature as well as the agitation. So we have good treat uniformly for the planting material. Then we validated, here we can see the complete eradication of smut by this treatment, as compared to the control. Here you can see two blocks, simultaneous blocks, till harvest there was no infection. But here this is 100% incidence in the same plot, same planting material we used for this treatment. Then this is with respect to GST. Here also we validated in plant crop. In plant crop, here compared to control, you can see the effectiveness in plant crop and this tritone crop and in the field conditions also. It is only with respect to hot water alone. Compared to here we can see the difference in the improvement of germination and enhanced seedlings from the GST infected sets. Then this apart from hot water alone, we have combined with the nutrient treatment also. It was found to be highly effective in having the GST management. This one we can see the effective. Then this is the validated for different varieties. This is one of the variety. Then we have proved the validated this one. And this shows the different varieties efficacy of hot water treatment combined with the nutrients. Then we also found that this is found to be highly useful for varietal rejuvenation. The recently whatever varieties we are releasing in the country, before going for release, we are rejuvenating the variety. We are passing through the hot water and followed by the chemical treatment before releasing the recent varieties. Then hot water followed by fungicides. We found that there was no yellowing till maturity. And also it was in hybridization program, it was found to enhance the flowering. And this is the settlement facility we have created in our institute for all the experiments throughout in every year we are following. This is the demo plot for farmers purpose, nursery unit also. And apart from this, now we entered into the interinstitutional program. We have we are collaborating with the 6 ICAR institute for validating this technology for other crops. That is like, apart from sugarcane, potato, banana, tapioca, turmeric and ginger. So now we are collaborating with all the institutes. Then this we can see the efficacy of different treatments in banana, along with both hot water as well as
present our work. And also I must congratulate and appreciate the organizers to include the food safety in the theme. Most of the time we talk about food security, but we often ignore food safety. So I must congratulate the organizers to inclusion of food safety as one of the important focus area in the theme. So here what we are doing at ICRISAT, I'll give you a snapshot, especially on this particular problem. And so just before going into my presentation, just a minute about our institute, that is ICRISAT. Those who are not aware of ICRISAT, ICRISAT is an international crops research institute for the semi-arid tropics. Established in the year 1972, there were two foundations played a key role, Rockefeller Foundation and Ford Foundation, in the establishment of ICRISAT in India, with the huge support from the government of India in 1972. And we are a non-profit, non-political organization, agricultural research for development organization, and we have a mandate to work in the semi-arid tropics of Asia as well as sub-Saharan Africa. And we work on 6 mandate crops, especially 3 cereals and 3 legumes. The cereals include sorghum, pearl millet, and finger millet, and the legumes include chickpea, pigeon pea, and peanut. And we have our global headquarters in India, in Hyderabad, in Telangana, and then we have very strong presence in several countries in Africa, both in the East and Southern Africa region as well as West and Central Africa region. So this is briefly about ICRISAT. And before going into my presentation, I just want to give you another update. I don't know how many of you heard about this incident which happened exactly a year ago in India, last October 29, 30, 31st. In the central part of India, in Madhya Pradesh, there were 10 wild elephants entered into one kodo millet crop area, and they fed on the crop. The crop is heavily infested with a fungi, and a lot of mycotoxins were there on the grain, and within 3 days, 48 hours, 72 hours, 10 elephants were died. So this has become a big national issue, and had a discussion in the Parliament of Government of India, and they constituted an investigation team, and I'm one of the part of the investigation member, and several samples after the post-mortem received to my lab, and we found that huge quantities of cyclopyrazonic acid found in the gut of the dead animals. Cyclopyrazonic acid is also one of the important mycotoxin, often ignored, and produced by the fungi like Aspergillus, Penicillium cyclopeum, and other Talaromyces and other fungi. So this particular incidence underscores the importance of addressing mycotoxin contamination in food commodities. So I'm not going to discuss about this particular issue because we just started working on this issue, and our first investigation paper published last week only. This got approved in the Journal of Wildlife Science. So coming to the topic, actual topic, aflatoxins in agriculture commodities, and this is one of the world's most important qualitative issue and affecting many crops. A few important ones are like groundnut, maize, and several tree nuts. And first came into limelight in the year 1960, especially in the United Kingdom, when hundreds thousand poultry birds were died after fed with contaminated meal which imported from Brazil, peanut meal. And then the work started. Know what was the problem and what was the death of the poultry birds. And by the 1966, the researchers able to address that. The reason is the aflatoxins produced by a fungi called Aspergillus flavus. That's why the name has come, aflatoxin, Aspergillus flavus producer toxins. Of course, later there were many toxins were identified, but these are the few important ones: B1, B2, aflatoxin G1 and G2. B stands for blue fluorescence and G stands for green fluorescence because they have the kind of fluorescence property when you observe under the fluorescence light. And the question is, and aflatoxin M1 is also an important, especially the animals when fed with contaminated feed, the milk is also getting contaminated by aflatoxin M1. So the question is about what is this issue? Is it an economic issue or a health issue? So when, especially in developed countries like in the United States, they spend millions of dollars to ensure that the public safety as well as to ensure that the value chain is clean and no contaminated food and feed enters into the chain. So it's an economic issue for them, but that was not the case in especially in developing countries as well as less developed countries in Asia and Africa. So it has become a health issue in all these countries. So if you look at the effects of these, not just the health effects, it is also affecting hugely the trade. Actually, the trade prospects are also getting affected by the contamination of aflatoxins in food commodities in groundnut. And very recently, there was an evidence that all these years, liver cancer, hepatocellular carcinoma, one of the predisposing factors is aflatoxin contaminated diet exposure. But gallbladder cancer is also linked to the aflatoxin contaminated diet. That paper has been published very recently from India 2025. So you can access that paper and read. I'm also part of that investigation. So if you look at the regulations worldwide, keeping in view of its negative health as well as trade effects, several countries, more than 100 countries in the world are imposing stringent regulations on these aflatoxins. European Union is one of the most stringent, the list and the countries list imposing very severe regulations. Any commodity which is more than 4 microgram per kg, they won't allow for direct consumption. And there are other countries which are having 10, 15, up to 20 microgram per kg. So just this is one sharp snapshot of India's peanut exports. If you look at India's peanut export scenario, India is unable to export peanuts to the European Union just because of this particular reason, very stringent regulations of aflatoxins. We could able to send our peanuts to other countries where slightly lower regulations were in place, like Indonesia, Vietnam, Philippines, but our exports to European Union is almost nil. So even though European Union is one of the big niche market for quality products, but we are unable to export because of this particular issue. So if you look at what are the most important predisposing factors for this problem, especially in groundnut, I would like to focus on groundnut. So the first and foremost reason is the end of season drought, and then the soil health issues, and then the insect damage as well as the nematodes and termite damage, and the improper drying and storage after harvest, and also the varieties, which are all important predisposing factors for this particular problem. And the interventions can be divided into two, primary and secondary, but we don't talk about secondary interventions, only we focus on the primary interventions, which are agriculture interventions. So we at ICRISAT, we work in several dimensions to address this particular problem: host plant resistance, biocontrol aspects, and also some of the agronomy practices with which we can able to reduce this particular contamination. But overall, not only at ICRISAT, globally, the limited success has been noted especially in case of host plant resistance. Even today, we don't have a kind of varieties which are highly resistant to pre-harvest aflatoxin contamination. So with that, actually, we started working on one unexplored area, that is seed coat, because there are two important physical barriers to enter the fungus into the groundnut kernels. One is the shell, and the second one is the seed coat. So the fungus has to cross these two important physical barriers to enter into the kernel and then produce the toxins near the cotyledon. So we want to focus on the seed coat, and we want to see what kind of biochemical composition is helps to stop the fungus growth as well as toxin accumulation. So here we have selected two important varieties. One is known as resistant after several field trials, that is 55-437, and one is highly susceptible, that is TMV2, and we want to deeply understand what kind of biochemical composition differs between these two varieties. So when we first started this experiment almost 6-7 years ago with the help of USAID Peanut Innovation Lab project, so in the TMV2, when we remove the seed coat, so when there is no seed coat, you can see the full of fungal growth. When there is a seed coat, even though it is a susceptible, the fungal growth is limited actually. So the same in the case of 55-437, which is known resistant variety. When you remove the seed coat, you will see the fungus growth fully, and when there is a seed coat, intact seed coat, you will see very less fungal growth on the seed coat after challenging with Aspergillus flavus toxigenic strain as well as non-toxigenic strain. This clearly giving us a kind of glimpse that yes, there is something there on the seed coat, and which is stopping the fungal growth as well as its proliferation on the seed. And also you can see many-fold changes when compared to the susceptible variety in terms of the fungal DNA present on the seed. So the second experiment is we thought of looking at the soluble and insoluble peanut seed coat biochemicals from these two varieties after challenging with the toxigenic one. And again, we found very interesting results that there is a significant differences between the resistant and susceptible one in terms of extracted soluble and insoluble seed coats which are inhibiting the fungus growth. And important thing is seed phenolic compounds. We started looking at the seed phenolic compounds present in these two varieties, and there were several phenolic compounds which was significantly different, but one which attracted to our attention is the ferulic acid, which is significantly high in the resistant one compared to the susceptible one. And there was some literature showing that ferulic acid is an important fungal growth inhibitor. And then we started looking at the ferulic acid. When you go on increasing the concentration of ferulic acid twofold, threefold, fourfold, we have seen a tremendous inhibition in the growth of the fungus. So that means this particular phenolic acid is playing an important role in the seed coat in stopping the fungal infection as well as toxin accumulation. And also we have looked at the phenyl propanoid related genes, which have shown again significant expression pattern differences between these two. And one of the most important thing is the lignin monomers actually. So there was no significant difference among the total lignin content between susceptible and resistant one. But there is, if you look at the lignin monomers, H-lignin is one important one. There is a significant difference between these two varieties. That means again lignin is also playing an important role in stopping the fungus growth as well as toxin accumulation. So overall, we found that the intact seed coat protects and inhibits the A flavors colonization, especially when we understood both the resistant and susceptible one. And also there are several other evidences which are supporting our hypothesis that seed coat is very, very important in stopping the fungus, so that we can look at the seed coat, how we can strengthen, how we can manipulate the biochemical constituents so that we can reduce the infection. And further, we even identified some of the important genes, candidate genes, which are linked to this particular resistant one. And also the secondary cell wall thickness is very, very important. As I mentioned, the lignin deposition is playing very important role in the case of resistant one compared to the susceptible one. And clearly you can see the histochemical analysis, the more and more deposition of lignin corresponds to the less and less inhibition by the infection by the fungus. So, and then we need to look at we need to look at the developmental biology of the seed coat. That is very, very important, because the seed coat is not going to be formed in 1 day or 2 days. Actually, there is a huge process, takes weeks of time for the formation of seed coat. We need to better understand at what time this lignin deposition is happening, so that we can able to look at the manipulation of this particular seed coat. And then we also work on several other aspects, not only host plant resistance, like water management is very, very important one, like how best we can able to keep the soil field moist for a longer period, especially before harvest. And then you can see the mulching plays an important role, which keeps the soil moist for longer duration and thereby reduces the pre-harvest aflatoxin contamination. And then we also work on storage aspects, especially drying and storage. And then the drying methods always important. The storage is one I want to highlight is hermetic storage, which is really gave us very good results in stopping the mold fungi infection as well as insect damage during the post harvest, because of hypoxia and hyperparvia conditions. When you store your groundnuts under in these kind of hermetic storage bags, this is one. Purdue improved crop storage bags, PICS bags, we have been promoting these bags after thorough evaluation. We found that they are very, very effective in keeping the quality of groundnuts. And this has been published in paper. And the diagnostics is another important one, because most of the diagnostic tools that we are currently using in the laboratory are cumbersome actually, because you need to bring the sample to the laboratory and you need to crush the sample, you need to extract the toxins to quantify, either using chromatography methods or immunoassays. But there is a need for the non-destructive and rapid methods of aflatoxin destruction, which is need of the hour, and the research has to be now focused in that direction, especially for the thing. And few other important areas are, like yesterday Dr. Andy Kumar mentioned about microbiomes. That is also one unexplored area, especially to address aflatoxin contamination. We need, rather than focusing on one or two important biocontrol agents, we need to holistically understand the microbial community structure and its role on the pre-harvest aflatoxin contamination. So that's also one unexplored area, and we need to look at it. And another thing is about biochar and other some soil cake soil amendments is also we need to better understand how it can reduces the populations of these toxigenics. So that's all, and I thank my our especially donors who supported our work, USAID, but unfortunately USAID Innovation Labs has been closed starting this year. We were unable to take this work to the second phase. Yeah, thank you. Yeah.
So thank you very much for the very clear, and you finished, I think you are the first one since the beginning of the workshop to have finished before the deadline.
I reduced my slides a lot yesterday night.
No, thank you. Thank you very much. No, it was an important thing, and if I try to make a link with what is happening in France, I think for peanuts, who are not very strong in peanut production, but or groundnut production, but we do have the same problem for maize production because of climate change, where there are more and more hot summers, and also in some areas humidities that favors that. So before asking the audience if you have any question, I had one question. Do you understand the ecological processes of the pathogen responsible for aflatoxin production? Because often when we try to understand the mycotoxin production processes, it's something that is not clear. Yeah. So I was wondering if you have some ideas about what are the conditions for aflatoxin conduction, especially focusing on the ecological balance between microorganisms.
Yeah. So what we understood, of course, the globally is Aspergillus flavus is not an aggressive pathogen. It's a soil saprophytic fungi, and especially when the soil microbial diversity is compromised, when the microbial diversity is very low, and then this particular fungus flares up. And also the temperature niche is very wide for this particular fungus. It can even thrive well above 40 degrees centigrade in the soil. So in that conditions, most of the beneficial microbes, they may not thrive well. So the diversity will go down, and then this particular fungus will flare up. So that is one of the most important thing. That's why we always mention that the microbial diversity is very important in the soils. As long as the soil health is good and very rich in microbial diversity, you can suppress this particular Aspergillus flavus populations in the soil. Yeah.
Thank you. So do we have any questions?
Yes.
Yes, one here, one there.
Yes.
Thank you. For it's not actually question, it's maybe the house participant clarification. So what's the level we would call it as resistant, or maybe is recommended for consumption of the aflatoxin level, or what the level we should not consume, or that's the threshold level would like to inform.
So in India, if you talk about India, so the Food Safety Standards Authority of India, FSSAI, is the official body, so which imposes these regulations. And it was earlier 30 micrograms per kg was the limit, but now recently 2018-19 onwards, they reduced it to 15 micrograms per kg. So safe limit, allowable limit, and any commodity which is up to 15 is allowable into the food chain.
Thank you for a very precise presentation. So,
yeah, but again, of course, it may not be harmful in 1 day or 2 days. It's a chronic exposure. That is very important. Acute exposure you may not see actually. But if you get exposed to the contaminated diet over a period of long time, 10 years, 15 years, 20 years, and if your immune system is compromised, especially in patients where liver cirrhosis and other hepatitis viruses, and then those are the vulnerable population get affected more.
yeah, thank you for your good presentation. I'm here.
Yes, sir.
I think for the groundnut, the soil-borne diseases, we are recommending the biocontrol agents like trichoderma
Yeah.
and of course the bacteria is there. So is there any interaction studies between the trichoderma species as well as the Aspergillus flavus?
There were many papers published actually in the literature. You will find many papers. Trichoderma is one of the important biocontrol agents even to address aflatoxin contamination too.
Okay. Is there any, the trichoderma also produce the secondary metabolites, is it?
Yes.
Is there any adverse effect on the groundnut?
Yeah, but we don't know the harmful nature of those metabolites. But
that's what I want to know.
Yeah.
Because trichoderma also equally produces the secondary metabolites.
Recently I saw one paper that the trichoderma also a pathogen for post-flowering stalk rot in maize, a paper published from US, Bangalore, Dr. Harish. All these days we have been recommending trichoderma to control post-flowering stalk rot in maize, but they identified that trichoderma is also one of the causal organism for the post-flowering stalk rot in maize.
So Aspergillus flavus also in the maize.
Yes.
So that interaction input I want. So if it is not there, I think if you come across one or two papers, it will be interesting to read.
Yes.
Okay. Thank you.
So, with your permission, so,
yes, sir.
So I have a couple of, rather my ignorance only. It's for the audience also. The aflatoxin producing strains. Do they really cause any disease in groundnut or maize?
There was one disease like
but what I'm asking is the one which is really producing aflatoxin.
No.
You mean to say that aflatoxin is no way connected with any kind of virulence on the plant?
Yield reduction, no, absolutely no. Because when you want to call it a disease, it should cause some yield losses. But there is absolutely no yield losses in peanut.
That means there is no way it is causing any disease in the plant per se.
Yes, absolutely.
That opened up another discussion what my co-chair asked. The ecology of this aflatoxin production is still a mystery. Is there any precursor in the soil system which promote this aflatoxin synthesis?
There are two actually, of course. The research is saying that under moisture stress conditions in the soil, the root exudate produced by the peanut is aggravating the reactive oxygen species ROS is aggravating the biosynthetic pathway of Aspergillus flavus. There are like 25 genes involved in the biosynthetic pathway of aflatoxin, and there is some trigger but not fully known. But this reactive oxygen species in the soil under specific conditions, high temperature, low moisture content in the soil is triggering the biosynthetic pathway. And also another important is the calcium. So usually in the soil, calcium acts as an inhibitor of the biosynthetic pathway. So when there is no enough calcium in the soil, and then the biosynthetic pathway happily, it takes away and then produces the end product, that is aflatoxin.
Okay. One more small interaction I would like to have from you. So I happened to guide one student from Cameroon, and he worked on aflatoxin in corn, maize. And his study was very interesting study. He compared the population of soil associated aspergillus versus the grain associated aspergillus. And the statistics, the paper is published in Scientific Reports now last year. It's a very interesting finding. The soil population of aspergillus is dominated by non-aflatoxin producing strains, but the grain associated aflatoxin is dominated by aflatoxin producing strains. So what's your comment on that? Because we were unable to explain why there is a more population of aflatoxin producing aspergillus on the grain when compared to the soil.
Maybe the substrate.
Yeah, then another discussion we got into that. But it is yet to be tested. Is there any link with the insect damage in those grains and the aflatoxin biosynthesis?
Absolutely, because that is what we have been telling for the past many years. Fungus as such, they don't have an aggressive pathogen, they don't have the mechanism to break the cell walls. And then they always look for an opportunity for any insect damage. Entry points, they use those entry points and enter quite easily. So that could be possible. Insect damage is always encourages the fungi to quickly enter and then produce the toxins.
Yeah, that opened up another discussion. I think Bharadharan is also sitting here. He is working on aflatoxin for many years. And the hypothesis right now is aflatoxins are produced to deter the insect rather than the plant.
Of course.
And because aflatoxins are toxic against the animal system, not the plant system at all. So maybe that's a new area of line. Somebody can open up. It's a nice presentation.
Thank you.
Any more question from anybody? Otherwise, you'll wind up this presentation.
this is both pre-harvest as well as post-harvest problem. So there are some people who argue that this is only a post-harvest issue, but that's not correct actually. It even happens during pre-harvest also.
So let's give a big hand to Hari.
Thank you.
So with that, we will move on to the third presentation this morning. The third presenter is Nagendran Krishnan. Dr. Nagendran Krishnan graduated and postgraduate and doctoral degree from TNAU in Coimbatore. And he also had a part of his PhD program he spent in Washington State University. Then subsequent to that, he joined in Agriculture Research Service and appointed in Indian Institute of Vegetable Research, that's somewhere in north, I think for the benefit of French team. It's in Varanasi. And right now he has been transferred to Banana Research Institute in Tamil Nadu. And that's 3-4 hours drive from Coimbatore. So he has traveled from that institute now. So over to you, Nagendra.
Thank you, sir. Thanks for the introduction. Good morning, chairs and delegates. Now I'm going to present on this managing viral disease of banana to ensure the food security. Actually, before that, I would like to give a brief introduction about our institute, National Research Center for Banana. It was started during 1993 at Tiruchirappalli. And we are maintaining nearly 540 germplasms of banana. In that, that includes 117 germplasms of exotic lines also, and some wild bananas also we are maintaining. And we have developed some bioreactor technology. Over that, we are multiplying the tissue culture banana plants, and we are commercially supplying to the farmers as well. And we have developed several post-harvest technologies and crop protection technologies as well. So this is the global, now coming to the presentation. Banana in global scenario, banana is a very important crop, not only in the context of domestic consumption and also in the context of export. It is a major crop after rice, wheat and maize, and it is being cultivated in nearly 135 countries with the production of 163 million tons. And nearly 400 million people are feeding this banana. So it is ensuring the food security as well as the for the farmer's point of view, it is giving the very good income generation. And nearly 1/4 of the global production of banana comes from the India, and major consumers are from India and China. And next, when we come to the Indian scenario, the banana total area it is being cultivated in 1 million hectares with the production of 37.47 million tons and productivity of nearly 38 metric tons per hectare. In 1991 it was just around 20 metric tons per hectare, but in 2024 it has increased to nearly it has doubled 38 metric tons. It was because of the interventions of the tissue culture technology. It has revolutionized the banana industry in India, and major producing states of the India are UP and Maharashtra is the leading producers, followed by Andhra Pradesh and then Tamil Nadu, and other states are also contributing the considerable amount of banana production. When we see the export, it is also being increased fourfold from the year 2018 to 22. It is increasing every year because the farmers they are getting more awareness, and they started exporting to get the more income. So it is a very important export crop as well. And though we are exporting and we are producing so much of banana, so many biotic stress constraints are being faced by the banana growers, which are caused by this fungal disease, bacterial, viral, insect pests as well as nematodes. Among them, viral diseases are very important, and nearly 8 viral diseases are infecting the banana crop globally. Among them, 4 are considered as the major viral diseases in India. That is banana bunchy top virus, then mosaic disease caused by cucumber mosaic virus, banana brack mosaic disease, and banana streak disease. Others are also there, but they are not that much important, and they are not causing the yield loss. First, when we come to the banana bunchy top virus, it is a babu virus. It is transmitted by the aphids. When the virus infects, the plant become bunching appearance, as the name goes, bunchy top. So the top portion of the plant become bunching, and it won't produce any yield. So it has recorded 100% yield loss, and several farm they have stopped cultivating because of this bunchy top disease. They have abandoned the crop. Next, when we come to the banana brack mosaic disease, this is a potty virus. It is also transmitted by aphids, and as the name goes, the brack mosaic, it is causing the mosaic pattern on the bract, which is the spindle shaped lesions on the bract, and not only on bract, it will produce the spindle shaped lesions on the leaf, and as well as the pseudostem and peduncle, leaf sheath. As some in extreme condition, it will produce the streaks on the fruits as well, which is later turns into necrotic, which fetches a lower market value. It has recorded the yield loss of 30 to 70% yield loss has been recorded from this. Next one is the banana streak disease. This is a paroretrovirus group. Actually, this is having the nature that it will, during the infection, it will integrate into the genome. There are two different genomes are present in banana, that is A genome and B genome. When they are B genome containing varieties are there, it will, during the infection, it will go and integrate. When the stress comes, then it will again come outside, and it will produce as a virus, as we are getting the infection of herpes virus in the humans. The same way it will come. So in a plant, if it is infected, it will produce streaks on the leaves, and later that yellow streaks will turn into dark brown and then black, leading to the drying of the entire leaves. Some banana cultivars are being cultivated only for the leaf purpose. So in that, this is a very major problem. So when the leaf gets infected with this one, they won't fetch the market value. And this is also very important virus in India. Next one is the cucumber mosaic virus. This is not restricted only to cucumber. This is virus infecting more than 1,200 crop species. So it is also transmitted by aphids, and it causes mosaic symptoms on the leaves, and if it is extreme conditions, the entire plant becomes stunting, and also causing the hard leaf, which is the youngest leaf, it will become rotting, and the plant won't yield any of the fruits. And these recent days, though it was reported in 1950s, in recent years, for the past 1 or 2 decades, it is emerging as a major problem in the major banana growing areas of Maharashtra and Madhya Pradesh, and in some extent in the Tamil Nadu also. Now, for the managing the viral diseases, I have categorized under the 6 different headings, that is quality planting materials, host plant resistance, biotechnological interventions, chemical methods, biological and cultural methods. In this, first we will see the quality planting materials. So the banana viral diseases are being transmitted through different modes. That is, insects also transmitting, aphids are there, and mealybugs are transmitting the banana streak viruses, and some farm implements during the intercultural operations are also being transmitting. But the major problem is from the planting materials. So this is a vegetatively propagated crop. So when the planting material is infected, it is spreading the virus infection from one field to another field. So the major culprit is the planting materials. Two types of planting materials are being used in the banana. One is suckers and the tissue culture plants. Both are spreading the viruses. So if we see the now as I told that planting materials are from COMs as well as the tissue culture plants. If a COM is infected, single mother plant is infected, it will spread the virus into one plant to 2 plants. If it is through the tissue culture, one plant, mother plant will be multiplied into 1,000, so it will be spread to 1,000 plants. So that much serious it is. As the technology also, tissue culture has revolutionized the productivity. In the equal way, it has increased the virus spread also. So why? Because there are 3 problems are there. Latency is there, virus titer is there, seasonal variation in the symptom expression. Because of this, when we select the mother plant, we could not able to get the virus free plants. So this make the spread of the viruses. So to address this issue, one study was they made that in 10 different varieties we have studied. In all the 10 different varieties, the latency was observed, and when the artificially inoculated plants, even up to 450 days of artificially inoculated, they are not producing the symptoms, but the virus are there in the PCR samples. So, and even the latent plants, they can be able to transmit. Even 3 to 6% of transmission was also being observed. So, to address all these issues in India, the NCS TCP has been taken up this tissue culture industry, and who are all producing the tissue culture plants through in India, they should get the certified from this NCS. They should accredit it with this NCS TCP, and through that they have to follow all the protocols developed by this one. So when they go for tissue culture multiplication during the mother plant selection, as well as when they distribute the plants infected, secondary hardened plants to the farmers, they have to certify test the plants for this virus indexing, and it should be certified that they are virus free. Then only they have to distribute the plants. So with this they can able to reduce the plants. So the virus indexing they are mainly doing through PCR, ELISA and RT-PCR assay for this BBTV, BSM, MOV through PCR, and CMV and banana BRAC mosaic virus through ELISA and RT-PCR they are doing it. Though so only these two methods are there, now they have approved for this NCS TCP. Several other methods are also been there for the virus indexing purposes. This virus indexing, the virus diagnostics, it should ensure this this 8 criteria that is assured. That is the abbreviation. That is assured means A stands for affordable, S for sensitive, specific, and it should be user friendly, and it should be rapid and robust, and it should be equipment free, and it can able to be delivered to the end users. It should be deliverable. So though there are different criteria, different categories of indexing diagnostics are there, it should ensure these things so that we can use it in the NCS TCP. If it is some are highly precise, they are costly, then we can't able to afford for the large extent. So, so different methods from our laboratory we have developed for the indexing of banana viruses, that is simplex PCR as well as multiplex PCR, and for detecting all the 4 viruses, that is quadruplex PCR also we have been developed in the single assay. And for the quantification, we have developed for the real-time PCR assay, which have identified even from the vector population also, and some extent this banana BRAC mosaic virus we have identified it in the seed, but there is no that much significance of this seed in seed transmission of banana BRAC mosaic virus in banana, but in banana. But this is also one study we have found out with this, and so from our laboratory we have developed several indexing techniques for which we are following through for the indexing services, which are based on the ELISA as well as the PCR and LAMP RPA based assays. Through this we are regularly we are indexing the private company samples also, and we are earning the revenues as well. And see, with this indexing, so one success story I would like to mention here. Virupakshi is a one banana variety which is widely, which is a very good sweetest variety, which is being cultivated only in the lower Palni hills. It is a GI tagged banana, and there they were growing across 18,000 hectares in the hilly area. But due to this introduction of banana bunchita virus in 1970, it has been reduced to 2,000 hectares, because in the hilly area the problem is that the vector population is high. We won't find the vector population in the plain areas, but because of the cool climate the vector population is high in the hilly areas. So because of that, spread was fast, and it has manage the entire crop. So to conserve this Virupakshi variety, that is GI tagged banana, a federation has been formed in 2006 in collaboration with TNAU and our institute to supply the virus free plants to the banana growers. So with that we have developed the mother block of banana virus free banana block in our institute, and through indexing and all, and we have supplied the mother plants to the private companies, and we have supplied it to the farmers. They have supplied to the farmers. So now the area has been increased to 10,000 acres in the Palni hills. So it is through indexing we have able to recover this extinct variety. The same way another I said that banana streak virus, it is also causing a problem in the leaf bee genome containing variety. So from our institute, over the 15 years of experiment, we have selected a line which is not endogenously present, I mean which is not integrated into the plant. So that kind of selection we made, and we have identified one line, and it is being circulated to the farmers, and it is giving a very good yield without any virus infection. For past 15 years they are free from virus, that is this banana streak virus. And though there are several advantages are there with the I have mentioned about this tissue culture plants. We can get the uniform growth, not only the virus free, we can get the so many other advantages such as the uniform growth, and they can produce the uniform bunches. Next we will go for the next heading is the host plant resistance. So the host plant resistance, very few germplasms are having the resistance for these viral diseases, that is banana bunchita virus and only BBRMV were identified, but though they identified, they are all not in the commercially cultivated lines, they are in the wild lines. So we could not able to explore, because in banana breeding is very difficult. So only propagation is through vegetative, breeding is very difficult in this, so it may not be explored. So with this disadvantage, biotechnological interventions such as the development of transgenic banana lines using the hairpin constructs, some attempts were made by targeting the replicase gene as well as replicase genes for the banana bunchita virus in Rastali variety as well as in the Virupaksha variety. They have developed, and it is working very well. And the same way for banana brack mosaic virus also, targeting the replicase variety, they developed a hairpin construct, and they in the Nandran another cultivar of banana cultivar, they have done it. And from our laboratory also, we have developed the transgenic Virupaksha line targeting the coat protein gene, and the plants are through bioreactor technology we are multiplied, and it is being evaluated. And multiple virus resistance transgenic construct targeting CMV, BBRMV and BBTV were also been developed. And some now the emerging technique, that is CRISPR-Cas9. In this we have targeted the viral genome, because the streak virus they have targeted the viral genome because streak virus is the endogenous and integrated genome. They have targeted it, and they have achieved the resistance for this particular virus. But utilizing this particular biotechnological intervention in the practical point of view, so many biosecurity issues, biosafety issues are there, so it may not be fruitful. So next one is the chemical options, chemical methods. Only this vector, wherever it is present in the hill area, this cool climate, we can go for this managing the vector, but in case of plains it won't work. Managing the vector, it may not be working in the banana as well. So some chemicals, imidacloprid and astaprid, they are using it. In addition to that, some other predators and parasites are also being used for the managing the vectors. One thing careful thing, since the vectors are hiding inside the crevices of the plants, so they should remove all the trashes, then only they have to go for spraying the particular chemicals. And not only for managing the vectors, we can to destruction of the plants in situ. It is very important, because when we disturb the plant, it may spread the vectors may spread to one place to another place. So we have to destroy the plants in situ by injecting the herbicides. The dosage of herbicide is very important based on the girth of the plant. For every 5 centimeter girth of the plant, we have to inject 1 ml of herbicide that we can able, that is only sufficient to kill the particular plant. And in a single clump, how many plants are there? Each and every plant, they have to be injected accordingly. And next one is the biological methods. Biological methods from our laboratory, we have studied it through the Bacillus and Pseudomonas application through root feeding and soil drenching. They have increased the plant vigor and they have delayed the symptom expression when we apply before the virus infection. Once the virus is infected, after that we are treating, there is no much difference at all. So pre-virus infection, we have to go for this one. And next one is the cultural methods. So several cultural methods I have highlighted, and another one is that including the indexing and vector management. So another one is the fertilizer management also plays a major role in virus disease management. So in case of brack mosaic, from our laboratory study for the managing the brack mosaic virus, they have increased the dose of recommended dose from 125% and 150%. It has equally the infected plants has produced the yield that is 16 kg per bunch has been produced in disease plant as well as a healthy plant by increasing the dose. So, but the same was not reflecting in case of cucumber mosaic virus. When we increase the dose, the same 125, 150, it is not working here. So, it based on the only 2% increase has been seen here. So, in conclusion, the virus-free planting material is the cornerstone for viral disease management, and preventive approach is very important, and we have to go for an integrated way. It should be farmers should be educated, and in collaboration with the institutions. Thank you.
Okay. So Nagendran, you finished just on time. It's very good. So still 19 seconds left. Yeah, over to audience for any kind of clarification. Sir, please hand over the mic.
Thank you. Okay, I suppose that all these viruses you spoke about were about on banana for export or banana for desserts, and I was thinking, what about plantain? Is it the same?
Both are same only.
Situation?
Yeah. Banana, it includes both banana as well as the plantain.
Okay, so all these viruses are also present in plantain? Okay, and plantain and the other one, I don't know how you call it, the dessert, they are grown at the same place together?
Yeah, here in Kerala they are cultivating this plantain called a variety called Nendran. They consume for the, as such they will consume. By cooking they will consume it. And some other varieties are fruits are also there.
Okay, thank you.
Yeah, thanks Nayendran for nice presentation. I wonder whether the CMB is transmissible from vegetable crops to banana and vice versa?
Yes sir, it is transmissible sir.
So from banana to cucumber
yes,
and other
going,
vegetables
sir.
also?
Yeah, it is going sir.
Okay.
In case of Jalgaon, that is a major banana hub in India. In Maharashtra state, there both the crop which are nearby banana, they are growing cucurbits. Both are having this viruses are there, but weeds playing a major role in that case. So there is no strain difference at all, sir. There.
Okay. Okay. Thank you.
Thank you.
Yeah, I don't see any students are in proactive interactions. So these are all an opportunities. He comes once in blue moon, you know, bringing French scientists and bringing scientists from all over the country. Students are simply sitting like a mute spectator. Is not expected actually. So you discuss among yourself, and some students should ask some question from the next presentation onwards. Okay, that's my observation. Yeah.
Maybe you can consider me as an old student. No, I had a question because banana also is produced in tropical territories of France, and actually it's an important question because we have a scandal, I don't know if you've heard about that, which is a chlordécone use, which is an insecticide that lasts at least 700 years and has tremendous effect on human health. So now it's very difficult for the population, not only farmers, but also the population to grow vegetable in their own garden. So I think you never used chlordécone, which is great. And a few years ago in the French West Indies, there was a program, an experimental program, where colleagues tried to design cropping systems based on banana using zero pesticide. So I was wondering, do you have such initiative in India trying to use zero pesticide, no herbicide, no insecticide, no fungicide? And if not, do you think it would be feasible?
Any comments on that?
There is no such initiatives.
No such initiative, but this point convey to your director to have such a program. So I have some observations. If no more questions, yeah, students, very nice, yeah. Somebody got his. Yeah. Stand up and ask, please. That's the way you should ask.
Sir, apart from the virus diseases, for the post-harvest disease management, what are the advanced technologies, sir, like anthracnose?
Yeah, from our institute study, they have identified some of the yeast-based biocontrol agents. So they are working very well, but now we have not yet formulation has not yet came. It is in evaluation stage, and biological control-based methods we have developed it. and from chemicals, the pre-harvest spraying, it is also reducing. So
Essential oils.
essential oils, it may not be practically it may not be applicable. I mean to say that it may not be affordable, and
But in many research papers like basil, they have used the basil, lemongrass and thyme oil like essential oils, sir. So in India, is it there any such kind of
the
practice?
researchers, they are going in our institutes also, other scientists, they are taking up the research on this essential oil as well, but I was not aware about those results.
Thank you, sir.
Yeah, I think it's a very relevant question. Post-harvest pathology in banana is a hot topic, because yesterday also we talked about it, post-harvest losses, and banana is one of the victims of that. So certainly we are fair in terms of controlling the diseases in the pre-harvest in banana. The successful technologies are there in the country. And Government of India launched a very good program way back in 2002-2003 time, sanitizing the banana suckers from viruses through indexing programs. I think that program is one of the most successful program in the country, where the viruses delivered in the farmer's field is significantly reduced. So this is one of the success stories in banana, how science can lead to clean planting material. But the post-harvest is still a hot topic. It's my suggestion rather, we should really focus more on post-harvest, especially the anthracnose.
Yes, sir.
Right? And her observation is very true. And some students in IRI, they work on this same aspect. The oil from high-value crops like clove and whatever, thymol, it is being tested, but nothing is in commercial. What I understand. For the information of French group who is sitting here, in banana, worldwide, we have some serious problems, especially in Central American countries, moco wilt. I think those who are working on bacterial problems, he must be knowing. Up to now, we have not recorded this disease from India. So from that perspective, India is in a very good position in terms of the banana production. So moco wilt is one of the dreaded diseases in banana. So far, it is not recorded from India, right? Number one. Number two is, very recently, there is a new disease outbreak in Tanzania and some of the North African countries, Xanthomonas bacterial wilt. Yesterday there was a presentation on Xanthomonas. So that disease also we have not yet recorded from our country so far. So from that angle, perhaps the Indian banana is also in a kind of a very nicely placed, because these two diseases are having the potential to devastate banana plantations world over, right? So that's in addition to whatever you presented. Okay. But I have only one question. 2 months back, I received a call from Jain Irrigation from Maharashtra. Jalgaon, yeah. There is some serious outbreak of some sort of bacterial infection in bananas. Yes, and I suspect it's Erwinia. So what's your comment on that? Whether any investigation is going on?
So Erwinia, we are suggesting only the management is through bleaching, application of bleaching powder, sir.
Whether the pathogen is proven beyond doubt?
Yes, sir. They have proved the pathogenicity as well.
Yeah. So I told.
Dr. Soumia. From here. So Dr. Soumia is an associate professor in the Department of Plant Pathology at the College of Agriculture. So from what I understood, it's a small village, Padanakad, right? Padanakad in Kerala. And you are specialized, she's specialized in the management of pathogen in general and viral diseases in particular. And you will be focusing your presentation on the management of viral disease on pulses, which I notice is a very important part of Indian food. We have many dal and so on, so it's an important topic. Thank you.
Thank you, sir. Respected Chair, Co-Chair, and delegates, good morning all. As sir had told just now, I am placed in College of Agriculture, Padnakad, but most of my service I was worked in Krishi Vigyan Kendra actually. So most of my experience is field experience, but while working in Krishi Vigyan Kendra, we had PG students working under the guidance, and they were working on plant virology. The work was being conducted in the small molecular biology lab we had at RARS Patambi.
Laptop support output laptop
Coming to pulses, as we all know that in India it plays a very important role in ensuring food safety as well as in bringing sustainability in agriculture. These pulses, they are very rich in protein, and it is a cheapest source of protein, and Indian population being mainly vegetarian, they play an important role in satisfying the protein needs of Indians. And also pulses form a very important part in cropping system because of its importance in fixing this atmospheric nitrogen and adding organic manure, fertilizer to the soil, thus reducing the usage of fertilizer, and thereby also a way to reduce this climate, to improve this climate resilience. In that way also we can have these pulses find a way in agriculture. So particularly in Indian situation, it is very important crop, and government has been promoting pulse production through various programs, through Krishi Vigyan Kendras and all. We had National Food Security Mission, under which we were having this cluster frontline demonstration in pulses. So all these were going on otherwise also, but now India is focusing more on promoting pulse production, in which this Atmanirbharta scheme in pulses, where our aim is to produce around 35 metric tons or 50 lakh tons of pulses annually.
Touch screen. Touch screen. So we were seeing that the importance of pulses in Indian scenario,
And it is also an added economic benefit for the farmers, because from, if we convert that yield into an equivalent yield of rice, we can get from 1 hectare within these 2 or 2.5 months, they can get around additional yield of, when we convert it to equivalent yield of rice, it will come up to 2 tons per hectare. So, since, even if it is this much important, and we are the major producer of pulses in world, and we are producing around 25% of pulses in world, still our domestic demand is not met, and we have to import pulses around sometimes about 18% of pulses have to be imported. And to meet our protein requirement, but present our availability is 43 gram per person, and it is, actually it has to be increased to 55 gram per day. Because of that, government is more focusing on pulses. Atmanirbhar scheme in pulses aim to have 350 lakh tons annually. For this, we have to increase the pulse production annually by 2.2%. When we see the productivity of different countries and compare it to ours is very much low when compared to other countries. It is having 2.5 times greater yield recorded in other countries. Our yield gap come to 220 to 602 kg per hectare, and several biotic and abiotic factors are contributing to this bottleneck. Mainly these biotic factors, weeds, pests and diseases contribute to the yield gap, and we have diseases alone, we have 10 to 15% yield loss annually. When we see major pulses grown in India, chickpea is covering around 35% of the area, then comes PGNP, green gram, black gram and lentil are the major pulses, and then other pulses are also coming. When we see the diseases, loss caused by diseases, we can see that from 10 to 100% of yield loss has been reported in various crops due to diseases. Among this, viral diseases also play a very important role. We have several viruses reported in pulses, either in field condition or in experimental condition. More than 50 viruses have been reported, which is belonging to several genera, Bigomo, Tosco, Kukumo, Boti, and all are reported. The major viruses in pulses in India include this chickpea chlorotic dwarf virus in chickpea, which is having this reddening side symptoms in Deshi type and yellowing type symptoms in W type standing, and also phloem discoloration is seen. In PGNP sterility mosaic virus is an important virus where the whole plant will become sterile when infected at an early stage. Mainly yellow mosaic viruses are the major constraint in green gram and black gram production, which is a Bigomo virus. This is transmitted by white fly. And groundnut bud necrosis virus, the Tosco virus, is also a major problem in cowpea, bean and green gram, and it has been in field many cases mixed infections have been reported, and this will lead to more yield losses. Woodbine leaf crinkle disease, though still now not assigned to a particular virus group, it is generally considered as a viral disease. When we consider viral disease, it is capable of incurring up to 100% yield loss it can cause. Hence, it is very important to manage these virus in pulse crops. In Kerala, mainly the pulses are green gram, black gram and cowpea. These pulses only we are having. Other pulses, PGNP and all, we will have as a border crop only. Chickpea is not grown in Kerala. And Kerala, my student has worked on this yellow mosaic incidence. We had around 12 to 100% yellow mosaic incidence was noticed in Palakkad, Malappuram area with the yellow mosaic leaf stunting, leaf reduction, etc. as a symptom. And electron micrograph has found geminate particles, and PCR with MYMV and HYMV primer had shown positive results, and which was aligning with the sequence was aligning with the MYMV isolates, and in the phylogenetic analysis they were falling well into the group where Tamil Nadu viruses are reported. Definitely it will be the neighboring state, and so it should fall in that category only, and those isolates which were amplified using this host gram yellow mosaic virus was falling into a definite other distinct category. Other viruses reported from Kerala University from Kerala itself, reported in cowpea, black eye, cowpea mosaic virus, tospo virus, and cowpea aphid-borne mosaic virus are reported. Coming to the management aspect, I have categorized it into three. That is, one directly, either directly focusing on the virus, it is all but still I have classified it as directly targeting the virus and preventing the transmission of virus, and also improving host plant resistance. Some antiviral principles has been reported in some botanicals such as Variolae diffusa, Bougainvillea spectabilis, and all these have shown reduction in disease in field also. We had got around a 70% reduction in port culture, but when it comes to field, it is getting reduced to 38 and 34% reduction. And Philandus neruri and Variolae diffusa against black eye cowpea mosaic has also been carried out in Kerala Agriculture University. It is also showing around 60 to 90% reduction under local lesion host. Then there is nanoparticles, which is directly interfering with the viruses, either blocking its entry or its replication. It is directly interfering the virus, and there are certain research works going on silicon nanoparticles at College of Agriculture, Vellanikara, and at Kerala Agriculture University. This is under cowpea aphid-borne mosaic virus, and very promising results were obtained. Only thing is that these nanoparticles, we have regulation in using field condition, and this can also be used as a carrier particle for transferring these antiviral agents. It can have improved field level resistance. When we come to interfering transmission, we have to see how it is getting transmitted. So certain viruses are getting transmitted through seed. Mungbean yellow mosaic virus is also reported to have 32% seed transmission, and Mungbean leaf fungal, it is transmitted by 71% by seed only. So protection of quality seeds is very important since it starts from the seed. Vector management is also another aspect in which in field what we are adopting mainly like prophylactic spraying of system. Since most of these viruses are transmitted in a persistent manner, we can go for systemic insecticides for the management. But when it comes to pulses, of course it is grown as a low input crop, and farmers won't be adopting such a prophylactic insecticide spray always. So here also we can go for nanopesticides. So it is host plant resistance will be more sustainable option in pulses for plant virus disease management. And certain works have been carried out in Kerala Agriculture University. We had Pseudomonas fluorescens inciting resistance response in plants. Then again the Centophyte Bacillus pumilus against tospo virus, and Piriformospora indica against black eye cowpea mosaic virus. There are several resistant varieties developed at IIPR and also this Pulse Research Center at Vamban. All these varieties, and some of the varieties are popular in Kerala also. In green gram we have this Virat variety, then IPM 2-14 Shreya, then Tamil Nadu variety CO8. These ones are in Kerala field also, they are showing resistance. In black gram we had this Vamban 6 to 11 varieties. This 6 it has already shown breakdown of resistance in field. Vamban 6 is now showing a mosaic symptom, whereas Vamban 8 is being popularly cultivated there. So all these indicate that it should be a continuous process, and we have to go on releasing these varieties because virus will be changing the strains, and they will be overcoming the resistance. So it has to be a continuous process, but through conventional breeding techniques it will take much time. It can be assisted with molecular tools. Some genome editing tools can help this. This RNAi interference has been utilized in managing MIV in cowpea by certain other researchers. And this is another that our such transgenic approach has been very much improved with the introduction of this CRISPR-Cas technology, where we can directly focus where to integrate this gene, so it will be giving more better results. Such types of work is also being taken up in this is a work from ICGB, and all these have regulations. When we comes to transgenics, in field application we have several regulations in India particularly. We have this topical application of double stranded RNA. This can be more easily adopted if developed into a field level technique. This is a work from our Karthikeyan sir's lab only. Such works are also being carried out. Mainly the SCOT protein and repeat protein are being used for such type of RNA construct formation. So this is giving very good result. The thing is that we have to take it up to the field level. So to conclude, we have several viruses reported in pulses, and it can cause up to 100% yield loss. And as far as management is concerned, it is a houseplant resistance is a sustainable option for bringing management. And topical application of double-stranded RNA can be effectively utilized. And we have to take these technologies to field level so that the farmers are benefited by the technology. We have several technologies have policy regulations, so we have to think of ways to overcome that. Thank you for listening. Let us efforts bring smart points.
Thank you very much for the nice presentation, and you deal very well with the technical problem and the timing. So thank you for that. Do you have any question? Yeah, I was thinking maybe you might have a question.
Yes, maybe I didn't follow well when you were speaking about seed transmission. You have a slide showing that you treated seeds with imidacloprid. Seeds were treated with imidacloprid.
No, that is
and I didn't understand well why.
It is an insecticide to. It is a systemic insecticide. It is a recommendation given by one author. Actually, I think for initial stage itself, for defending that virus, they have given it as seed treatment, and that treatment they have recommended as the best practice.
It is to prevent whitefly.
Yes. For managing vector.
It's not for seed transmission. It's not because of the seed transmission.
Seed transmission, no. For combating the vector transmission at the initial stage.
Okay.
Because in most of the cases, initial stages, they are causing more damage. So they have given the recommendation as seed transmission, not for seed transmission.
Thank you. Another question?
Thanks, Samir. I'm very curious to know about the VPM 8 status because it was released during 2018, if I'm correct, 17. But last couple of years, we are seeing the symptoms, though it was released as a total resistant variety. But what is the status in Kerala?
Kerala actually VPN 8, it is widely grown out. I mean, it is not very popular, as you know, it is green gram and black gram, not very widely grown, but at least in Palakkad it is grown as a third crop in many regions. Mamban 8, I have not noticed actually, sir, will say. Mamban 6 it was broken down in 2017-18 itself, but Mamban 8 I haven't noticed.
Thank you.
Thank you. Any other question in the audience? Yes? Sorry? Okay. So it's time to make a break now. So we have to. Okay, sorry. One last question. Okay.
Yeah, it's the same question like, is it really important in Kerala, the pulses? It's known for plantation crops, especially coconuts, spices.
Yes, sir. But we have paddy also.
Of
so
course,
it is very
paddy.
well suited crop for as a third crop in paddy fields. It is really remunerative for farmers also.
Yeah, paddy, we know that in Palakkad area, to some extent.
Because sometimes when paddy fails, they want to resort in this.
Yeah, that's all I wanted to ask. Thank you so much.
Thank you very much. So we can thank you very much. Okay, so now we have a coffee, tea, masala chai break. Anything? How long do we have, sir? Sorry? 15 minutes break. Okay, so let's come back in 15. Thank you.
Initial discussion. We'll do that. Share my details. It's very nice. A few weeks ago, we went to the dog because we were struggling. Yeah, we are happy to collaborate. And it's great to do this. Which language is used? I speak comfortably English. I was from New Delhi. The last one was the last low pressure. Yeah. So I do have students from Canada. My group. Only no other language.
எப்போ இல்லை ஆனால் இருக்குது 11 o'clock தான் சொன்னாங்க 11 தான் சொன்னாங்க இந்த எல்லோ
அவங்க வர மாதிரி போட்டு விட்ருங்க பேசாமல்
நான் ஓப்பன் பண்ணி வச்சுருது
ரெண்டு ஆன்லைனோட முதல்ல இது பண்ணி விட்டுடலாம்.
தலைவர் இப்படி தெரியுது. நல்ல வேலை பார்க்கறதுல மண்டை உடைக்கிறது. இங்கேருந்து போதாட்டிருக்கு. நான் கொடுத்துருக்கேன்ல, அதனால நினைக்கிறேன். நம்ம சிஸ்டம் தான் இங்கே வருது.
கீழே தான் ஆப்ஷன் வாங்க. அவங்க பார்த்து சொல்லுறாங்க.
நேற்று இங்கே ஒரு ப்ராஜெக்ட் பேப்பர் வச்சுருந்தேன். இங்கே வச்சுருந்தேன். நேற்று இன்னொரு பேப்பர் வச்சுருந்தேன்.
பென்
இருக்கா? இல்லை இல்லை இல்லை, நேற்று நான் ஒரு நம்பர் வச்சுருந்தேன். எங்கே போகுது? பார்த்தது நல்லா போச்சு, பார்த்தது நல்லா போச்சு.
ப்ரெசன்டேஷன் முடிச்சா மோடி எல்லாத்தையும் மாற்றணும்.
இல்லை இல்லையே கண்டிப்பாக மாற்றணும் இல்லை இல்லை பண்ணிக்கலாம். வரும்போது இல்லைன்னு போட்டுக்கலாம். நல்லா போச்சு பார்த்தது. இல்லைன்னா இங்கே அந்த நேரத்துக்கு கையும் ஓடாது காலும் ஓடாது.
அவங்களோட ஐடியிலே போயிட்டீங்க.
ஆமாம்
அவங்க ஜாயின் பண்ண மாட்டோம்.
மறுபடியும் எல்லாம் வருது. Don't ask. It's a challenge. பண்ணலைங்க சார்.
பண்ணலையா?
At the end of the session, I would like to make a remark because I saw that in many presentations there is a problem with some statistics.
Okay.
So can you give a general advice?
Yeah.
I don't know, but I don't know if it's good or not.
சுஷில்
ஆன்லைன் கனெக்ட்
ஜாயின் பண்ணியம்மா
இன்னும் பண்ணி
Yeah.
it's not on one.
கனெக்ட் ஆகிட்டு பாருங்க.
இல்லை வரல பார்ட்டிசிபென்ட் ஜாயின் பண்ணி தான்
நான் பார்த்தேன். ஃப்ரான்ஸ்லேருந்து ஒருத்தவங்க கேட்டாங்க, அவங்க தான் 11:30 கேட்டாங்க. அவங்க வரல. அவங்க கூட
யார்
சுஷில்
யாரும் தெரியல நீங்கள் 2 பேரும் இதான் தந்துடுறீங்க.
நாங்கள் ஃபோட்டோ வந்து பாருங்க.
அங்கேயே பண்ணிக்கலாம் சார்.
அதான் ஓகே.
பேர் வந்து
SUSHC கால்
இல்லைங்க சார்.
சரி இதை
இல்லையா
சரி கூகுள் அவங்க வந்து
Shall we start? Okay. Then in the meantime, can we take a moment? Welcome back all delegates. We will start the next presentation. The next presentation is delivered through online mode. The presenter is Dr. Sushil Kumar Sharma. He is a senior scientist at Indian Agriculture Research Institute, New Delhi, the same institute from where I am just coming. Sushil Kumar Sharma has done his doctoral degree from Indian Agriculture Research Institute. He is specialized in plant virology. He has got a lot of expertise in plant virus detection, diagnosis, and viral disease management. He is using the next generation sequencing technology to understand the viral etiology of croplands, and presently handling externally funded projects funded by NASF, that is National Agriculture Science Fund, DBTs, and other funding agencies. So with this brief, yes, I think he's a recipient of several awards that has been already highlighted in the handout that's given to you. With that, I'll request Dr. Sushil Kumar Sharma to deliver his lecture. It's 18 minutes, Dr. Sushil, and the sooner you complete, more time we get for discussion. Try to wind up the presentation in 18 minutes. Over to you, Dr. Sushil.
Respected Chairman, Dr. Vanti sir, I hope I am audible.
Yeah, you are very much audible.
Yes, okay. So, very good morning to everyone. Most respected Honorable Kumar sir, the chair of the session, respected delegates, co-chair and everyone. So, I'll be sharing my slides and then we will discuss. Hope my slides are visible, sir.
Yes.
Okay, thank you. So the topic assigned to me is on application of new techniques in diagnosis and management of emerging plant viral infections in different crops, and I'll be taking examples of mostly horticultural crops which I am dealing with. There is some issue in slideshare. So can you see the second slide, please?
Yes.
Okay, so this is how I will be talking. The flow of my presentation will be mostly will discuss on viral diseases, their significance in agricultural cropping system, understanding their molecular epidemiology and also etiology, how infections can be detected through different techniques including high throughput sequencing, particularly unbiased detection, and how this information can further be taken forward to develop the specific targeted detection tools and which can then be used for management. So as far as viral infection of different cropping system is concerned, the ICTV master list as on 24, the latest one which is updated, which is edition of 30 and 39th edition, it recognizes around 1,677 species which infect plants, and they are classified to 119 genera. So this basically specifies that how many distinct group of viruses across the domains which can infect the plant species, including agri and horticultural crops, which are the crops of our interest. Now viruses, while they are so diverse, they have different modes of transmission, and they can be transmitted through vegetative propagation, they can be transmitted through insects, nematodes, fungus, even mites. They can also be transmitted through parasitic plants, contaminated foods, seeds, pollens, and also through root contacts as well as soil and so on. So this makes their management very difficult, and as far as our country is concerned, we have vast array of the viruses infecting both annuals and perennial crops. If you to mention here, you can see Indonesian leaf curl in okra, you can see windy yellow vein mosaic virus in windy infection, you can see recently emerged important viral disease in temperate region, that is in apple. You see necrotic mosaic virus, you have different infections of Piscagian virus like agents on citrus and so on. So similarly, a lot of infections you see under Indian conditions, including both annuals, field crops and perennial crops. So why such infections are very important? They are very important because they causes a lot of losses in terms of yield, quality and productive life of the plants. In annuals, in the same life cycle, you see the losses in the yield and quality, whereas in perennial crops, the losses are even more pronounced because Once the infection happens, the plant remains infected throughout its life cycle, and that viral load accumulates over the year, and it also acts as a source of initial infection for further spread to the planting material or new stocks which are planted. As far as the losses they cause in terms of the yield, particularly in perennial crops, many such studies has been conducted across the globe, both at economic level as well as yield losses in the percent, and you see that many infections like in apple, banana, you see infection up to 90 or sometimes 100% can be observed, and those can be correlated to the losses in terms of the yield. Now, when we understand the viral infections, it is very important to diagnose them. And now, initially, we were diagnosing them at a particular species level, but now the concept has changed to the viral complexes, and mostly the diseases which are caused by the viruses are not because of one viral infection, but it could be because of mixed infection by many of the viruses. So as you know, their complete cure is impossible, so it is always important that we diagnose them well in time and we take the preventive measures, not for them to spread further in the plant population, and also we take control measures in terms of producing the clean stocks and keeping it clean, whatever way we can keep through certification or regulation. So as far as their diagnosis is concerned, we can have either non-targeted method or we can have targeted method. When we say non-targeted method, it could be through symptoms, it could be through transmission, which can be undertaken through biological means or through indicator host. It could be through structural characterization of the viruses. Generally, the most common is electron microscopy, which we do. So you can see whatever viruses are present in a particular sample. Nevertheless, many times our two methods, we miss many of the viruses because of the less load or less concentration in the tissue, or not improper sampling we do. So we have many other methods in the second line, which could be the targeted one, where we develop the specific diagnostic assays to detect them. So it could be through ELISA, which is a serological based method, or you can have any molecular detection method, which could be PCR, RT-PCR, isothermal assays, or so on. Very recently, we are now using, for last more than 1.5 decade, plant virology has witnessed a paradigm shift in detection of viruses associated with a particular disease, and we have high throughput sequencing, which is mainly employed for detection of the associated viruses. So we already mentioned that the other commonly used method have their own limitations in terms of specificity and sensitivity and limited coverage. So in the era of omics, we employ high throughput sequencing for the non-targeted neutral detection of the viruses, where we sequence the entire nucleic acid in a sample where we want to see what are the viruses associated. So high throughput sequencing has a specific advantage because here we do the parallel simultaneous sequencing of the multiple DNA fragments. Mostly you take RNA as a starting material in case of all viruses, whether they are DNA viruses or RNA viruses, because all viruses have their transcripts in the once they start replicating in a particular cell. So you can use the RNA, total RNA from the infected cell as a starting material, and you can convert it to cDNA and then followed by parallel sequencing on either of the platform. Mostly we use shotgun approach, and then lot of massive data you generate, and this data can be further analyzed to see what are the viruses present. So it basically provides a good advantage because you can identify the potential pathogens without having any prior information. You don't need any antibodies, you don't need any sequence information which is otherwise required in nucleic acid based detection method. Here you can detect everything whatever present in a sample which you use as a starting material. It is rapid, comparatively rapid, accurate and efficient, and you can see the entire virome. This is at the later stages that you correlate it with a particular infection and you correlate it pathogenically, but at the initial point of time you will be able to decipher what are the viruses which are present in a sample. The other advantage of this technique is that you can identify both known and unknown viruses as well as characterized as well as uncharacterized viruses, including understanding of their diversity, evolution, and also molecular epidemiology. Even a very low titer viruses can also be detected in the sample. That is again an advantage. So how we basically do, you take a plant sample, you subject it to high quality total nucleic acid isolation, and this DNA or RNA you take for preparation of the cDNA libraries. You label it with the adapters. Adapters are basically the known sequence labels, and then you make libraries and you sequence. Once the data is generated, you analyze it. So you analyze it in a way that you would like to see what are the viruses present in particular sample, and so here you need a massive data analysis tools, which is basically a computational tool using in-house or whatever validated pipelines. In most of our studies as well as globally, we use short read sequencing to analyze the virome of a particular sample, and this is generally done on Illumina, as you can see from the right side of the slide. That mostly Illumina gives you very good amount of data which can be further assembled to see the viruses. So how such techniques has been used by us to analyze the viruses? I'll take a few examples from our own studies. So we started working extensively on this particular technique, and based on our experiences, we have developed, validated, and optimized some workflows to analyze the viromes in a particular sample. So you can see mostly we take the samples, so it could be asymptomatic samples as well as asymptomatic samples which we want to see. You subject it to high quality RNA isolation. You check the quality of the RNA, then you go for the library preparation. Once the library is prepared, you subject it to
Sushil, you have 5 minutes to go, Dr. Sushil.
Only 5 minutes?
Yeah.
So high throughput sequencing, and once that data is generated, you analyze it to see the viruses. So using this technique, basically we have identified number of viruses and also understood their epidemiology. One of our study, which is published in Physiological and Molecular Plant Pathology, we studied the grape nurseries and both rootstocks and cymes, and undertook their virome studies to see the viruses present in them. So once it is done, we could identify at least 12 viruses present in either of the rootstock and cyme. Once such viruses are identified using Illumina platform, we wanted to detect their prevalence specifically in mother banks of rootstocks and cymes. So if you see this table, just to highlight the major findings, you can see that there were some viruses and viroids which were consistently present in rootstocks, whereas others were present only on the cymes. So basically it shows that how viruses have moved from the rootstocks to the cymes. In addition to understand their epidemiology, we were also able to identify some of the novel viruses like GLRABS, grapevine mitovirus, and one novel viroid, that is grapevine yellow speckle viroid 2. Similarly, in case of apple, we had developed a pipeline and we trained it on multiple data sets, and we could decipher the infection of new viruses, including the coco viruses, that is concave, citrus concave gum associated virus, and apple rubbery good virus, which is a rotavirus. So in the same line, we had deciphered the virome of the pear, identified the viruses associated with the symptoms which we observed in the field, We also deciphered the virome of the plum, similarly peach, and many more crops which we are doing now. So despite having this advantage of the high throughput sequencing, we can still have some issues. If you have a sample bias, means you don't sample proper tissue which should be subjected for sequencing before RNA isolation. So also degradation could be an issue, because if this technique is very sensitive, so contamination is again an issue as well as some bias. So once the viruses are identified using high throughput sequencing, we can take the viruses which are economically damaging and we can develop the specific diagnostics which can be used for targeted detection. So our group here, we had been using isothermal detection assays, mostly RPA, and it is a recombinase polymerase amplification assay. The assay could be performed at 37 degree isothermal temperature in 30 to 40 minutes. And using this particular assay, we have developed wide array of the diagnostic assays for detection of the viruses as well as virus-like agents, including those of lateral flow dipstick assay. So this one we recently published in crop protection, and this is for detection of Candidatus librivector asiaticus. And the advantage and beauty of the assay is that you can detect the infection in the crude plant extract, isolated, prepared in a specific buffer or particular solution. Similarly, we have developed our duplex RPE assay for simultaneous detection of 2 DNA viruses in banana, that is banana streak MY virus and banana streak bunchy top virus. So we developed a number of assays, at least for a dozen of viruses, and many of them now we are using under national certification system for tissue culture based plants. So with this, we have completed detection, both neutral and specific detection. I'll take a few minutes just to brief about the management, how we can use different recent techniques for management of viruses. So our group is also using topical or exogenous application of dsRNAs, which is in vitro synthesized for management of the viruses. And recently we have used this technique for management of one of important virus infecting chili, that is chili venom motor virus. So we use the dsRNA cocktail against 3 genes, that is replication, silencing, suppressor and assembly, and this application can prevent the infection of this virus in the plants. Similarly, we are also using CRISPR-Cas9 for editing of the potential or known susceptibility factors. So our group has started working on rice, and our senior group here in IRI, they have a long working experience on using this particular technique in tomato. So their target is pelota, which is one of the known susceptibility factor, and which helps the establishment of the viral replication in tomato. So this pelota gene, which is a susceptibility factor, at a particular site, that is position number 16, they used a CRISPR-Cas to mutate it from valine to the glycine, and once these mutants are developed, these mutants of tomato are resistant to tomato leaf curl viruses, particularly New Delhi virus and Bangalore virus. So this is just to conclude my presentation, that techniques like high throughput sequencing will help you to identify the viruses at a neutral level without having any bias. Once you identify the viruses, whatever important group of viruses are present, you can develop diagnostics, and then this can be used for regulation or certification program. As far as management of the viruses is concerned, we now have some new techniques like dsRNA based management as well as CRISPR, which can be used for management of viruses, and we can use if we have susceptibility factors known in a particular host virus pathosystem, and they can be managed. So I would like to thank all my funders as well as collaborators for for this work, and also TNU for inviting me to talk briefly on the work which we have been doing. Thank you.
Yeah, thank you, Dr. Sushil. That's a very nice presentation. So, over to audience if you have any observations, any questions, any clarifications.
Good morning. Thank you very much for the presentation. I was wondering, when you use NGS for viral characterization, do you have a quantitative readout about the abundance of the viruses? And if so, does that help you decide on whether the virus is a potential threat? Because this is also a related question. Is when you have a virus in the pen, does that necessarily mean it's a threat in terms of disease and yield limitation?
Thank you for your question. If I correctly understand, you asked that whether we were able to quantify it in a particular tissue. So yes, basically we had been using different algorithms to quantify mostly copy number, FPKM values in a particular sample, and then we were validating it with the qPCR. So we had done some studies on understanding the seasonal dynamics or viral load in these tissues which we maintained under insect-proof glasshouse conditions, particularly in citrus and grapes. So we definitely use to quantify the viral load using high throughput sequencing, not only detection, at least under for some of the crops of our interest. And sorry, I missed the second part of the question.
So the second part was, when you detect a virus, does this necessarily mean it's a threat, it's a disease that can be a problem, a limitation in cultivation?
Yes, so basically in most of our studies we used this technique to identify the viruses associated. Particularly recently there was a disease which emerged in our rice growing region, and that is dwarf disease of rice, and we used high throughput sequencing to identify the cause of it, etiology, and it was a Fiji virus, that is southern rice black streak dwarf virus. And then thereafter we took some biological studies to transmit it using its vectors and prove cause postulate. But in many of the clonally propagated viruses, sometimes our interest remains on understanding the entire virome too. So it is both.
Okay, thank you very much.
Thank you.
Yeah, there is a question from Karthikeyan.
Shashil, this is Karthikeyan. Thank you for your nice presentation. I have a small doubt to know, when you target the pelota gene in tomato, did you get any off-target effects in tomato?
So basically this is work done by Dr. Anirban's group, and they have a good lead, and off target of course they have tried to minimize. So they even use base editors to minimize the off targets, and only ensure that you have only targeted changes done in a particular genotype.
Thank you.
Any more questions from anybody? Okay, if no more question, it's a wonderful presentation, Dr. Sushil, and I know you have been doing good work, and probably if you have come here in person to interact with the students and others, I know why you are not here today. I think he's traveling abroad. So all the best. Yeah, thank you so much. Yeah, let's give a big hand to him here.
Yes, sir. Thank you. Thank you.
Okay, so it's time to move on. So maybe while I make a short introduction to you, Laila, can you set up your presentation? So Dr. Laila Bani Bail is a researcher from CIRAD. She's an ecologist. She's specialized in natural regulation and agroecological management of pests. She worked a lot
on cacao crops in several regions of the world. And actually, I'm curious to know what is our new research program, because she's working now in the Réunion Island. So we are in France, but still in the Indian Ocean. And also there is something specific to you, Leila, is that you have a good look with regard to the skin color of the people here in Coimbatore, and it's a pity you could not make it to be with us. Okay, so the floor is yours.
Okay, thank you very much. Thank you very much, Jean-Noël, and thank you to everybody for the invitation, and sorry that I'm not with you today. So I'm Leila Benibey, and I just make one slide to present you where I am now. So I'm in a research unit based in Réunion Island, in the southwest of Indian Ocean. And in my research unit, we work on plant communities and biological invaders in tropical environments. And so my research institute develops innovative control methods against pests, disease, and weeds in agrosystem, but also new ways of conserving indigenous forest environments. And we are making research and training in tropical crop protection and biodiversity preservation in terrestrial ecosystems. And we are composed of 3 research teams. One is worked on emerging pathogen genomics and epidemiology, another one on diversity and sustainable use of plant genetic resources in tropical environments. And my research team work on ecological dynamics in island environments. So now I work more on the regulation of ants in Réunion Islands. So because we have big problems of ants like pests in different crop systems. But today my talk will be more on what Jean-Noël said, what I did before. I mean regulation and agroecological management in tropical perennial crops. So I will present some work that I did in Latin America with my PhD student. And so my presentation will be very different from the ones that you just saw, because I would like to introduce some concept and to make a discussion about how to study this natural regulation, and then how can we make a link between regulation, agroecological management, and ecosystem health. So maybe that can be a little bit different. So I hope that will interest you. So maybe you know that coffee and cocoa are mainly cultivated in agroforestry systems. So we have almost 30% of cocoa systems and 40% of coffee systems in the world that are cultivated under shade trees. Sorry. So just to present you an agroforestry system with cocoa or coffee. So mainly you have like the arboreal compartment that will make shade in our system. You have the host plant compartment with a plant like coffee or cocoa. Then we have an herbaceous compartment and a soil compartment. And what we have also, we have farmers. Mainly the ones that I worked with were like small farmers. So they used to have practices, but mostly they don't really use pesticide, or if they don't have the choice, because they don't really have income to pay for pesticide use. So what they do mainly in these systems, they use to manage the biodiversity that is in the system, and then they have like agronomical practices with pruning or weeding. And for pest and disease, most of them used to harvest disease pods. So they just have a look at the plot, at the pods, and when they can see some kind of disease appearance, they just remove the pod. Whatever they can use or do to use it. So in the region where I worked, I will present you later, we have like 4 pests and disease. So on the left we have a Phytophthora, and then we have a cocopod borer that is present in Indonesia, for example, in Southeast Asia. Then we have Monilia, which is a frostopod rod, and then we have another type of Monilia, which is And these four pests and diseases can occur at the same time, sometimes on the same tree. And for example, on the left, we can see that one pod can be infected at the same time by Phytophthora and the Coccophophthora. And what we saw that in literature, we don't really have a very good definition of pest and disease regulating service. Some people say that there are all the biological and physical-chemical mechanisms within an ecosystem that help regulate the appearance, incidence, and severity of pathogens or pests known to infect and cause damage to other organisms within the ecosystem. But with my team, we prefer to use the capacity of living organisms to influence direct and indirect pathways that control the appearance, incidence, and severity of pests and pathogens that reduce the yield of crops. So the objective of this work was to know how does inclusion of biodiversity in an ecosystem influence the pest and disease regulating service. And first, we would like to know how to evaluate this regulating service in cocoa agropestry system, and then which are the pathways that biodiversity harness to influence the pest and disease regulating service. So first, we just have to know that there's no methodology that exists for accurately measuring the pest and disease regulating service efficiency. Because, for example, there's no integrated indicator, then most people use incidence, but sometimes the incidence is okay for one pest, but that's very hard to follow incidence of many pests at the same time. And also the problem that we can have is, for example, the severity is a very difficult indicator for farmers to follow, and it's not convenient for them, because when they can see severity of the disease, that means that the sporulation has started, for example, for vanilla. So it's too late to control that. So we propose to use the yield loss as an indicator. So what is yield loss? If you just look here, you can see that for cocoa, at the beginning you have a potential loss. That just means that this is the yield that is determined by factors like radiation, temperature, and it's very related to the genetic potential of the species. And then you have like the availability of water, nutrients that can limit this potential yield and arrive to an accessible yield. And what we have after that, we have occurrence of pests and disease and climatic disasters and weeds, and that will give you your actual yield. And the differential between the accessible yield and the actual yield is your yield loss. So we decided to maybe work more on this indicator to be able to evaluate the regulating service. And what did we measure to work on the yield loss? In the system, we work mostly, as I said, on the biodiversity compartment, so the arboreals, the herbaceous, and the soil compartment. We also evaluate the pest and disease compounds, the host plant, I mean the cacao plant itself, and that helps us to determine the attainable yield, and then we calculate when the pest and disease occur, what arrive for the yield loss and what is the actual yield for farmers. So for us, all these components of how the biodiversity is related to pest and disease components and what is the effect of the yield loss is the pest and disease regulation process. So we did this work in Peru, which is an important country for cocoa production, but there is also almost 60% of incidence of most damaging pest and disease on cocoa in the world. And farmers, as I said, they used to practice sanitary harvest, and they have a variety of cocoa that is auto-compatible. So we set up a protocol with different communities. We have sent 8 land plots, and in each land plot we have like a full sun and shaded plots. And we did different field missions during rainy and dry season. So my purpose today is not to present exactly our results, some of them, but just to explain you what were our, I would say that our workflow, our process of working, to try to be able to estimate this regulating service. So first we try to construct our indicator of seed loss. So as I said, we have a combination of disease per pod. So we collect the pods and then we open them, and we were just looking at how many seeds were losses and what was, if we have, and what was the status of the seeds. I mean, if we have just affected seeds that people can, for example, sell on the black market, if we have destroyed seeds that is completely lost, and if we have, and how many healthy seeds that we have. And we relate that with the incidents that we can see when farmers made a sanitary harvest. So then we make a kind of calculation, and we were able to estimate what was the affected seed ratio per pest and disease combination, the destroyed seed ratio, and the seed damage ratio per combination. And we also estimate for this particular clone what was the average seeds per pod. So what we saw is that, for example, for black pod, we have half of the black pods in blue that have affected seeds, and only 10% was completely destroyed. For example, for the frosty pod rod, we have around 70% that was completely destroyed, and 25% that was affected, and only 8% of the pods that was okay. I mean, you can use 8% of the seeds that are in a pod. So that was the first interesting result, and what we did with that, we combined that with the incidence at the tree level. So for example, now we know that with different profile, I mean, if the tree has only Phytophthora, or if the tree has Phytophthora and the Phosphorus, or if the tree has only Phosphorus, what is the expected yield for this particular tree? And then we use this indicator a little bit later, I will show you. And as I said, we also study the different pathway of how biodiversity can affect the yield. So we measured different compartments, as I explained. So first one was what was related to abiotic effect on the trees. I mean the visual deficiencies and the stress symptoms. And we have like a grid that we can explain if we have deficiencies in zinc, or if we have a solar stress, hydric stress, and other measurements. On the cacao tree itself, we did some type of architectural measurement with the height of the trees, the canopy, the height of the canopy, and many variables. We also measured the microclimate, since we know that the microclimate results from the effect of shade. So we made different type of measurement of temperature, of humidity. We also worked on vapor pressure deficit. That is a link between the capacity to maintain water for the tree, and this is a very good proxy for disease development. We measure the shape directly, so above cocoa tree and below cocoa tree, because the cocoa itself can make a shape that can impact disease occurrence. We measure also the soil chemical analysis, so we make some soil collection, and we try to see the potassium availability, nitrogen, the boron, the zinc, and the soil pH, whatever. We measure the respiration in the soil with different methods that are developed by some researcher in Iran. We also make like some visual evaluation of the soil. For example, the texture of the soil, if it was compact and if it's made by sand or other things, and also an evaluation of the composition of the soil. I mean, if we have for example, weeds, if this is bare soil, if we have branches, and other types of measurement. And what we did with that...
Just to let you know, 5 minutes left.
Okay, it's okay, completely okay. And so what we did with that, for this different compartment, we made some cluster analysis. So we have one compartment for architecture, another one for nutrient deficiencies, environmental stress, soil chemical, and the different measurements that I did, that I showed you. And we also have an attainable yield compartment. And what we saw that we have three kinds of clusters. One was like with low attainable yield, another one with medium one, and another one with high yield, high attainable yield. And what we did, we tried to find all the relationship between low yield, middle yield, high yield, with the different components of the compartments that we measure to find what are the conditions that favor finally this yield. And then what we did with that, I'm sorry, I'm quite rapid, but it's just to show you the process that we think and how we would like we wanted to make the analysis. Then we use like structural equation. Structural equation is interesting because that can help you to find direct and indirect effect on on the response variable. And what we did, we tried to explain what are the factors that influence the total shape, for example, architecture and associated shape, what are influencing also the soil cover, architecture, and total shape. So here is the part where we looked at the link between the different compartments, because as you know, shade tree will affect microclimate, will affect cocoa growth, will affect incidence. And then we try to find also the link with yield loss. So here we have a kind of results that are a little bit complicated, but just to show, for example, that associated shade has a positive effect on the total yield loss, and the total shade has a negative effect on opportunistic plant cover. So this is a really a systemic approach to try to understand what are the different pathways that will come and impact the total yield loss. So finally, for our first question about the evaluation of regulating services, we proved that yield loss be a variable integrated indicator. So maybe my talk will not show that exactly, but we have two papers that really put that in. This is a relevant indicator. And the yield loss framework combined with the pathway analysis allows us to evaluate the efficiency of pest disease regulating services in a kind of, how to say that, natural system when farmers come with a practice. And regarding the different pathways that we found that all the compartments have at least a direct or either indirect pathways, but some compartments like associated trees and the cacao risks themselves are very critical role. And the holistic view in the agro-system is really important, and the fact and also the combination of factors are important to be able to evaluate this regulating service. So as a conclusion, I say that, okay, we have many interactions, so I hope my talk helped you to see that we need to set up suitable protocol, to think about the statistical tools to use, and to need to, for relevant expertise of people from the soil, from pest and disease, to be able to set up this kind of protocol. The plant diversity and pest dynamic, as you know, varies according to the scale, spatial and temporal one. And we need to know how much biodiversity is needed to help farmers, how much shade should be used, what is the link with production, losses, and to better understand the mechanism, as I tried to show in my talk. And finally, I just want to have a question to see how we can make the link between agroecological protection and regulation services and the health of our ecosystem. So in my case, I'm in a cash crop system, so maybe we have two indicators that can be interesting, the profitability of the system and the sustainability of the system. And as you can see in the small graph, you have, for example, the ecosystem services, and maybe we think in our group that ecosystem services and the ecosystem health are very related. And for example, if you work with coffee yield loss, as I showed you for cocoa, you can see on this gradient of diversification that you have a trade-off between the coffee yield loss and the profitability of the system. And the profitability is the cost and the income that farmer has. And that can be interesting because with the cash crop, we know also that the livelihood of the farmers will depend on the money that they have. So this is something that maybe we can try to see how to go from ecosystem service to the ecosystem health. And I hope my talk helped you to try to think about that. Thank you very much for your attention, and sorry for being late.
Thank you, Leila. No, the timing was okay. Don't worry. Okay. I will ask if the audience has questions for you. I just have one. Because we saw that it was a very innovative way of trying to assess the quantitative natural regulation within agroecosystems, and I'm curious to know if you plan to adapt this methodology to your new research program on ants, and if at all possible, can you share on which crops will you apply this methodology if you plan to do so?
Yes, I'm planning to do so also because what is interesting with ants is that they are at the same time they can be positive and negative. I mean, they are effect in the system because sometimes they are good for the soil, but they are bad for and they are also able to do like direct damage. For example, here we work on potatoes and we work on also. How to say that, the pitaya, I don't know the term in English, but
dragon fruit.
And the passion fruit.
No, dragon fruit.
Yes, the dragon fruit and also the passion fruit and also on potatoes, because for example the fire ants is able to make some direct damage on this type of crops, and they are also mutualism with emitters. And in the system in Reunion Island, and this is also diverse system. I don't have the shade, but most of the time farmers used to have different fruits with different other crops, and all of that are just in the same plot. And we can see that sometimes that have an influence on not only on the presence of the ants, but sometimes also on the resources and the ants' behavior. For example, some of them sometimes are able to be more predators, and the same species in another system can be more, they look for sugar, so they are pest for the crop. So I would like to adapt this kind of approach to those systems to try to understand why sometimes the ants change their behavior, and how can we modify, if I can say that, the system to modify the ants' behavior and the effect on the crops.
Okay, thank you, Leila. And you know that for the positive part in Vietnam, for instance, some ants are used to protect crop production. So it's both sides of behavior. Yeah. Thank you. So do we have any questions to Leila with regard to this? Thierry?
Thank you for your presentation. I really enjoyed it. I was just wondering, so you are characterizing and analyzing some points to your analysis. So what's the size of the points? Is it just 1 tree or it's a bit larger? And I was wondering how many points do you need to characterize and analyze to have meaningful results with your approach?
Okay, so in my approach per plot, I have 40 trees. And and this is perennial crops, so and that's why at least I need 3 years of measurement of these 30 trees, and all the trees were followed up every 15 days. And then so we check for the different indicators of disease infestation, and the fact that we have the different trees helped us to. This is what was interesting in our work, that we can have a plot approach, but also a tree approach. And in what I presented, we work more at the tree level and not at the crop level, because we have only, 8 plots. And even if we try to find some similarity with plots, we are in a agroforestry system and almost all of them are different. So we work with 400 trees in our systems, and we try to find other proxy, like for example distances between trees, to be able to work at plot level, and to try to find the special patterns in one plot to try to have a kind of clusters of our trees, but that was very hard to do, and that was the approach at the beginning, and finally we decided to work at the tree level because the spatial clusters was not so easy to analyze.
Thank you. So if there is no other question, maybe it will be time to conclude. So maybe we can thank Laila. Thank you very much.
Thank you. Thank you very much.
Okay, and now we will have a short sum up. We prepared something with the two co-chairmen. Thank you.
Yes, thank you, co-chair. So, very engaging session. I think you would agree with me. We had researchers and scientists from 6 different diverse institutions in India and outside, starting from Sugarcane Breeding Institute, ICRISAT, University of Banana, Kerala Agricultural University, CIRAD, and IRL. Certainly they delivered their best. Just to sum up what we listened in the last 2.5 hours, the diseases of very important cash crops like sugarcane, of course food crop, banana, pulses, fruit crops from IRI, and tropical perennial crop like cocoa and coffee. And it was a very engaging session, and we learned a lot from these presentations and discussions. We also learned several technologies. The notable one is the hot water treatment, a successful technology developed by Dr. Malati and team in Sugarcane Breeding Institute. I think it's a lesson for all of us. What we do in basic science need to reach the society at large. I think here is an example today. A successful and very popularized technology for set treatment. It's very important because planting materials are the culprits. The subject ecology also evolved from one of the planting materials. I think if you sanitize the planting material, perhaps we need to avoid the problem. Prevention is better than cure, always. So I think that's a big lesson. I also learned a lot from Dr. Malati's presentation. We also learned diagnostic technologies depending on the CRISPR-Cas, RPA, different serological and molecular techniques. I think overall, it was a very engaging session. I think we learned a lot, and certainly the French team also learned a lot what's going on in India, and maybe this information would shape our future collaborations, anything we planned. With that, I conclude the summing up. And over to my co-chair, he has got some very interesting comments to make. I also agree with him. Over to you, Jim, yeah, please.
Thank you very much for this nice sum up of the session. Now, I just wanted to share with you one comment that I had since yesterday, actually, because in not each presentation, but in many presentations, we are dealing with either ratios or proportions or percentages, and these variables are a little bit specific because they are called binomial variables. You know, when you measure the incidence or the severity or the prevalence of a disease in the field, for instance, you can get 0 or 1. So this type of variable is specific, and often in the graphs that were presented, we didn't get any information with regard to the variability. So meaning, for instance, you go in the field, you take 30 plants, and you say, okay, I have 15 plants with some symptoms over 40, so the expected value of the incidence is 50%, but then you have no information of the variability. However, since it's a binomial variable, you have a functional relationship between the expected value and the variance of the thing. So my advice would be always try to remember that you can give an information with regard to the variability, even if you don't have replicates. Okay? If you have a sample size, then you can calculate that. So it was for the graphs without any information, but then we had graphs with some information. I think it was standard deviation or maybe confidence interval. However, they were calculated assuming that the distribution was normal. it's possible to make this assumption, but the size of the sample has to be quite high, and also the expected value should be a little bit close to 0.5. As soon as you get close to 100% or to 0%, you cannot assume anymore that it's a normal distribution. And in that case, there is another approach to propose confidence interval. It's called exact confidence interval. So the problem is that there is no equation that will give you the answer. It's only a numerical assessment that can give you confidence interval. You can choose any risk level that you want, like 5% risk level. And the key message is just try to remember that you can associate information on variability of your measurement even if you don't have replicates in your sampling strategy. And always think, do I have enough sample, and am I close to 0 or to 100% to make the assumption that the distribution is normal? Just a small methodological remark. With that, thank you very much, and I think we can enjoy a good beer with all the Indian products. Thank you very much.
Stay back, we listen to the announcement from the organizers.
We will break for lunch because there are 3 excellent sessions are going on in other calls. So our slot is between 12:30 to 1:30 for lunch in the dining hall itself. Move to the campus.